A single female snake can lay 80 to 100 eggs in one clutch, and somewhere in the Everglades right now, one of them is doing exactly that, coiled in the sawgrass, completely invisible, completely unafraid. Nothing in this ecosystem has taught her to be afraid. That's the actual problem. And the strangest part of the story is that the best weapon scientists have found against her offspring might already be sitting in your medicine cabinet. Since 2017, contractors, state agencies, and licensed hunters working across South Florida have removed more than 10,000 Burmese pythons from the wild. That sounds like progress until you look at the effort behind it. Removing a single python takes, on average, roughly 12 hours of combined search time. The annual Python Challenge, the state's flagship public hunting competition that draws hundreds of participants from across the country, typically nets somewhere in the range of 1 to 200 snakes per event. Meanwhile, nobody actually knows how many pythons are living in the Everglades. Estimates run from the tens of thousands into ranges wildlife agencies are reluctant to state publicly, because the honest answer is that the tracking methods available can't produce a reliable number. What researchers do agree on is that reproduction is outpacing removal, and it isn't close. The pythons didn't evolve here. They arrived through the exotic pet trade, most likely juveniles bought small and released once they outgrew what an owner could manage, which happens quickly for an animal that can reach over 17 feet and become a genuine apex predator. Researchers with the U.S. Geological Survey and the National Park Service have documented pythons preying on white-tailed deer in Big Cypress National Preserve, and mammal surveys across the python's range have recorded severe declines in raccoon, opossum, rabbit, and bobcat detections in the areas where pythons are most established. The Everglades gives them almost everything the Western Pacific or Southeast Asia never could in this abundance. Dense cover, warm water year-round, no winter to kill off a season's hatchlings, and prey that never learned what a python looks like. Every conventional method runs into the same wall. You cannot hunt what you cannot see, and a python's camouflage in sawgrass is close to perfect. You cannot trap an animal that isn't drawn to bait the way a raccoon or a coyote is. And you can't poison the water without poisoning everything living in it. So a small group of wildlife toxicologists asked a different question. Instead of trying to catch the snake, what if you made its food supply the trap? That question has an origin story that starts on the other side of the Pacific. In 1952, the brown tree snake arrived on Guam, almost certainly as a stowaway in military cargo. It's a mildly venomous, meter-long snake, not aggressive toward people, and for years nobody thought it mattered. What made it catastrophic was something nobody could have predicted. Guam's native birds had evolved for thousands of years with no ground-based predators at all. They nested low, they didn't flee from movement on the ground, and they had no instinct telling them a snake was a threat. For the brown tree snake, the island wasn't an ecosystem to compete in. It was an undefended pantry. Julie Savage, then a graduate researcher, spent years in the 1980s trying to figure out why Guam's forest birds were vanishing. She published her findings in the journal Ecology in 1987, definitively linking the collapse to brown tree snake predation, after finding snake-killed eggs in nest after nest across the island. By the time her work was accepted by the wider scientific community, the damage was already done. Guam has lost or effectively lost the vast majority of its native forest bird species. The snakes didn't stop at birds either, they climb. And Guam's power grid absorbed hundreds of outages a year from snakes crossing transformer lines, costing the island millions in damage and repairs. That crisis produced an unlikely tool. Researchers with the U.S. Department of Agriculture's National Wildlife Research Center discovered that acetaminophen, the same over-the-counter pain reliever found in Tylenol, is lethal to brown tree snakes at surprisingly small doses because the snake's liver can't metabolize it. You can't scatter pills across a jungle canopy, but you can place a measured dose inside a dead neonatal mouse, attach it to a small cardboard tab designed to snag in tree branches instead of falling to the forest floor, and distribute it by hand or by air. Field trials on Guam using this method documented substantial reductions in local brown tree snake activity in treated areas. It became one of the few tools in invasive species management that actually worked at meaningful scale. That success is what led USDA and University of Florida researchers to ask whether the same approach could work on Burmese pythons. Laboratory studies, published through USDA's Wildlife Research Program, tested acetaminophen-laced mouse baits on juvenile pythons and found the compound was lethal to them as well, with mortality rates climbing sharply as the dose increased. That's a genuinely significant finding. But it comes with a critical asterisk that rarely makes it into the retelling. This works specifically on juveniles, not adults, and the difference matters enormously. An adult Burmese python weighing well over 100 pounds has no interest in a mouse. It's hunting deer, not rodents, and a dead mouse in a tree means nothing to it. Juveniles are a different animal entirely. In their first year or two of life, young pythons are roughly the size of an adult brown tree snake. They actively hunt small rodents, and crucially, they climb through low canopy and understory vegetation, the exact zone where a bait mouse would be positioned. Researchers who've tracked juvenile pythons using radiotelemetry have described them as far more mobile and exploratory than the sedentary waterbound adults, moving constantly through vegetation in a way that puts them directly in the path of this kind of bait. The logic behind targeting juveniles is really a numbers game. A female python doesn't need to be killed to stop contributing to the population. She just needs to be prevented from ever reaching breeding age. If enough hatchlings and yearlings are removed consistently year after year across enough of a landscape, the adult population eventually stops replacing itself even without a single adult ever being touched by the program. To actually cover the python's range across South Florida, researchers estimate the scale required would be enormous. Millions of bait units, deployed repeatedly, not a one-time trial. And that's exactly where the plan runs into a problem that has kept it from moving past small controlled tests toward anything resembling a Guam-style rollout. Guam, by the time bait drops began there, had already lost most of its native bird life. The ecological math was brutal but simple. The system was already collapsed, so there wasn't much left to protect. The Everglades is the opposite case. It remains one of the most biodiverse wetlands in North America, home to hundreds of bird species, dozens of reptile and mammal species, and an entire web of predator-prey relationships that are still largely intact. Acetaminophen doesn't distinguish between a python and anything else willing to eat a dead mouse. Start with the animals already under pressure from pythons themselves. Small and medium mammal populations in parts of the Everglades have already declined sharply due to python predation, according to long-term surveys. A dead mouse lying in low brush is an easy meal for exactly the kind of scavenging and opportunistic species already struggling. And the damage doesn't stop at direct consumption. A squirrel or a possum that eats a sublethal dose can carry residue in its tissue. A hawk or owl that later catches that animal absorbs a secondary dose, and nestlings, with far less tolerance than adults, are especially vulnerable. These exposure chains are difficult to trace once bait has been dispersed across a landscape, which is exactly why researchers have been cautious about scaling the method up. The species drawing the most concern isn't a bird at all. It's the Florida panther. Florida Fish and Wildlife Conservation Commission estimates put the current adult and subadult panther population at somewhere between 120 and 230 individuals, confined almost entirely to a shrinking range south of the Caloosahatchee River, territory that overlaps almost completely with core python habitat. Cats, as a group, are unusually sensitive to acetaminophen. Their liver chemistry lacks the enzyme pathway that lets humans process it safely, and even modest doses can cause red blood cell damage and liver failure. Panthers are known opportunistic scavengers that will take an easy, effortless meal, and a population this small has essentially no buffer to absorb additional losses. That single fact is why most researchers who've studied this option describe deployment anywhere near panther range as an unacceptable risk, at least with current bait delivery technology. There's also a species most people have never heard of that makes the calculation even harder. The eastern indigo snake is North America's longest native snake, and it's one of the very few animals that actively hunts and kills young Burmese pythons. A natural check on the exact population this bait program is trying to suppress. Indigo snake numbers have already been reduced by decades of habitat loss, and there are active, funded recovery and reintroduction programs trying to rebuild them. Indigo snakes eat rodents. Whether they have any resistance to acetaminophen has not been established, which means the strategy carries a real risk of harming the one native predator best positioned to help. Aerial or hand-distributed bait isn't a precision tool either. Pythons favor habitat close to water, so bait naturally concentrates near marshes, canals, and open water, areas where a meaningful share of bait mice will end up in or near water and be scavenged by fish, turtles, and other aquatic species whose exposure is nearly impossible to monitor at scale. Florida isn't the first place to run this exact cost-benefit calculation. Australia's large-scale poisoning campaigns against invasive foxes, feral cats, and rabbits using Compound 1080 offer a useful comparison. The program worked koala and wallaby populations recovered in parts of Western Australia and predator control on Kangaroo Island protected native marsupials. But native eagles and other raptors died from eating poison carcasses, and some rabbit populations eventually began showing tolerance to the toxin, meaning the tool's effectiveness wasn't permanent. The broader lesson researchers have drawn from that experience is straightforward. No large-scale intervention in a complex ecosystem produces only the effects you intended. Collateral impact isn't a footnote. It's built into the method itself. Florida has also already seen how the public reacts to interventions like this. When the biotech company Oxitec received approval to release genetically modified mosquitoes in the Florida Keys as a way to suppress disease-carrying populations without pesticides, a method that had already achieved major population reductions in field trials in Brazil and the Cayman Islands, the local response was intensely negative, with organized opposition, lawsuits, and heated public meetings, even though the target was a mosquito that nearly everyone already wants gone. A campaign involving millions of medicated bait mice spread across public and private land, in an ecosystem home to panthers, eagles, pet cats, and endangered snakes, would almost certainly draw far more public scrutiny, not less. So where does that leave the actual response to the python invasion? Removal continues. Professional contractors and licensed hunters take thousands of snakes out of the population every year, and it matters, even though it isn't closing the gap on its own. Detection dogs trained to locate pythons by scent have shown real promise in controlled trials, finding snakes that human searchers walk right past, though scaling that up requires training infrastructure that doesn't fully exist yet. Radio tracking a captured female to locate breeding aggregations of multiple males has become one of the more efficient field tactics currently used by researchers. Scientists are also investigating pheromone-based lures and pathogen or genetic biocontrol approaches that could target pythons specifically without affecting native reptiles, though those tools remain largely experimental and years from field deployment. The acetaminophen bait strategy sits in an unusual place. It's one of the only methods with solid laboratory evidence that it actually kills the target species, and at the same time, one of the hardest to responsibly deploy at the scale the crisis demands, because the very features that make the Everglades worth saving, its density of species, its interconnected food web, are the same features that make broad chemical intervention there so risky. Researchers continue to study smaller, more targeted delivery methods that might reduce non-target exposure, but nothing resembling a Guam-scale rollout has happened in Florida, and current evidence suggests it may not happen for a long time, if ever, without a way to solve the collateral damage problem first. The pythons, meanwhile, keep breeding. The removal programs keep working, just not fast enough. And somewhere in the sawgrass tonight, another clutch of eggs is being kept warm by a mother with nothing in her environment telling her she has anything to fear. That's it for this one. If you want to see what happens as python range keeps pushing north out of South Florida, that's the next video, and it's worth sticking around for.